Sometimes when you really get into a topic, you realize just how much more complex things are than you thought.
Oh, totally. Like, you think you have a handle on something, and then boom, a whole new layer of complexity just hits you.
Yeah, and that's what today's deep dive is all about. It's about those times where a simple story about biology just completely falls apart when you look closer.
Right, and sometimes those moments lead to some really surprising discoveries.
Exactly. And today we're going to be talking about one of those surprises, and it has to do with aging. Specifically, the role of mitochondria in aging.
Mitochondria, yeah, those tiny powerhouses in our cells that are always being talked about when it comes to aging and energy and all sorts of things.
Exactly. So the basic idea, right, the
Yeah.
one that's been around for ages, is that as we get older, our mitochondria just sort of wear out. They don't work as well, and that leads to all sorts of problems.
Yeah, that's the classic narrative you hear about mitochondria and aging. Decline equals damage equals aging.
Right. But what if it's not that simple? What if there's more to the story?
Well, there is, and that's what's so exciting about the research we're going to be looking at today.
Okay, so what's the twist? What makes this research so special?
Well, it suggests that it's not just about how much our mitochondria decline with age, but when that decline happens.
And that's interesting. So it's not just about the severity of the decline, but the timing.
Exactly, and that's the really unexpected finding here. The timing of mitochondrial dysfunction might be even more important than the severity.
Okay, I'm intrigued. Let's unpack that. So where do we start with this deep dive?
Well, we're going to be focusing on a specific component of mitochondria called complex 1.
Complex 1.
Yeah, it's spelled out the number 1, not Roman numeral 1, COMPLEX And, it's a key player in the electron transport chain, that whole process that generates energy within mitochondria.
Right, the electron transport chain. I've heard of that. So complex 1 is like a crucial station on that energy production line.
Exactly. And to really understand the impact of its decline, the researchers turned to fruit flies.
Fruit flies? Okay. I know scientists use them a lot for genetics and stuff, but why are they so good for studying aging?
Well, for a couple of reasons. They have these relatively short lifespans, which means researchers can observe the effects of aging and genetic changes over a whole lifetime without waiting decades like they would with humans.
Right, that makes sense. So they're like a fast-forward model for aging.
Exactly. And on top of that, scientists have developed some really sophisticated tools for manipulating the genes and biological pathways of fruit flies, so they can really precisely control what they're studying.
So that's what these researchers did. They tinkered with the fly's complex 1 function to see what would happen.
Exactly. And they had some really fascinating results.
Okay, let's hear about those headline findings. What were the big takeaways?
Well, one of the most striking things they found was that even small reductions in complex 1 activity during the fly's development led to dramatically shorter lifespans.
During development, so you mean when they're still larvae growing up into adult flies?
Precisely. And that's already kind of counterintuitive, right? If you just assume that mitochondrial decline is a gradual age-related thing, then you'd expect that early disruptions wouldn't matter too much as long as the mitochondria are functioning okay in adulthood.
Right, you'd think the adult stage is where it really counts.
But that's not what they found. These small developmental hiccups had huge consequences for the fly's later lives.
So even subtle changes early on can have ripple effects that last a lifetime.
Exactly. And that was the first big surprise. The second one was even more unexpected.
Okay, now I'm really curious. What was that?
They found that severely depleting complex 1 in adulthood actually extended the fly's survival under certain conditions.
So you're saying they basically crippled the fly's complex 1 function and they live longer?
Under certain conditions, yes. And not only did they live longer, but they also seemed to be more resilient to various stresses.
Okay, now that is mind-blowing. So less mitochondrial function leading to longer life. It goes against everything we thought we knew.
Right. It really challenges the dogma that mitochondrial decline is always a bad thing for lifespan. It seems like in some contexts it might actually be beneficial.
So it's not just about decline equals damage. It's about how the organism responds to that decline.
Exactly. And it opens up a whole new way of thinking about mitochondrial function and aging.
Okay, so let's dive deeper into these findings. Let's start with that developmental impact. How exactly did they reduce the function of complex 1 in the developing flies?
They use a technique called RNA interference, or RNAi. It's a really powerful tool that allows scientists to specifically target and silence certain genes.
RNAi. Okay, I've heard of that. So they basically use this RNAi to turn down the production of complex 1.
Not exactly. Remember, complex 1 is a complex made up of multiple protein subunits.
Oh, right. So it's not just one thing.
Yeah, so what they did was use RNAi to target 2 different subunits of complex 1.
2 different subunits. Okay, so they were trying to see if it mattered which part of complex 1 they messed with.
Exactly. And the 2 subunits they targeted were ND18 and ND75. And these are actually really interesting because they have human counterparts that are involved in mitochondrial diseases.
Oh, wow. So they're not just random subunits. They're actually relevant to human health.
Yeah, ND18 is similar to a protein called NDUFS4, which is implicated in Leigh syndrome.
Leigh syndrome. That's a serious neurological disorder, right?
Yes, it is.
Yeah.
And ND75 is similar to another human protein called NDUX1, which is also linked to mitochondrial disorders.
So they're studying these fly subunits, but they have a direct connection to human diseases.
Exactly. And that's what makes this research even more compelling.
Okay, so they targeted these 2 subunits with RNAi, and they did this during development.
Right. But here's the key thing. They also had another group of flies where they only reduced complex 1 function in adulthood after the flies had fully developed.
So they had 2 groups. One where complex 1 was reduced throughout development and adulthood, and another where it was only reduced in adulthood.
Precisely. And this allowed them to compare the effects of early versus late complex 1 dysfunction.
And what did they find? What was the difference in lifespan between those 2 groups?
The flies with the developmental reduction in complex 1 live significantly shorter lives.
Significantly shorter. Like how much shorter are we talking?
It depended on the specific subunit they targeted and the conditions, but it was a substantial difference.
Wow. So even if the flies had normal complex 1 function in adulthood, that early hit during development still caught up with them.
Exactly. It had a lasting impact on their lifespan.
And this was true for both ND18 and ND75. So it didn't seem to matter which subunit they targeted. The developmental reduction was always bad news.
Right. It suggests that disrupting complex 1 early on, regardless of the specific subunit, is what matters.
But they made sure to control for the overall level of complex 1 reduction in adulthood, right?
Yes, that's a really important point. The researchers carefully measured the complex 1 activity in the adult flies, and they found no significant difference between the 2 groups.
So the amount of complex 1 reduction in adulthood was similar, whether the reduction started early or late.
Exactly. So it wasn't just that the developmentally affected flies had weaker mitochondria overall.
Right. The amount of damage was comparable, but the outcomes are drastically different.
Precisely. And that's what points to the importance of timing. The when is just as critical as the how much when it comes to a complex 1 dysfunction.
Okay, so that's the lifespan part. What about the stress sensitivity? You mentioned that the developmentally affected flies were also more vulnerable to stress.
Yes, they were. The researchers tested the flies' resilience to 3 different types of stressors: oxidative stress, thermal stress, and metabolic stress.
Okay, let's break that down. What do you mean by oxidative stress?
Oxidative stress is basically an imbalance between free radicals and antioxidants in the body. It can damage cells and is linked to aging and various diseases.
So it's kind of like rusting from the inside out.
Kind of, yeah. And to induce oxidative stress in the flies, they expose them to hydrogen peroxide.
Hydrogen peroxide. Got it. And what about thermal stress?
Thermal stress is pretty straightforward. It's just exposure to extreme temperatures.
So they basically cranked up the heat on the flies.
Yeah, they subjected them to high temperatures to see how well they could cope.
Makes sense. And what was the third type of stress?
Metabolic stress. And for that, they simply deprived the flies of food.
Starvation. So they were testing how well the flies could handle going without food.
Exactly.
And how did the developmentally affected flies fare in these stress tests?
They were much more sensitive to all 3 types of stress. They didn't handle the stress as well as the flies, where complex 1 was only reduced in adulthood, or the control flies.
So even relatively mild stress was a bigger deal for them.
Exactly. They were just less resilient overall.
And this increased sensitivity to starvation in particular seemed to be linked to problems with their fat body, right?
That's right. The fat body in flies is kind of like a combination of our fat tissue and liver. It plays a crucial role in storing energy and regulating metabolism.
So you could say it's the fly's metabolic control center.
Yeah, and the researchers found that the flies with developmental complex 1 dysfunction had significantly lower levels of triglycerides, which are the main form of stored fat.
So they weren't storing energy as efficiently.
Right, and when they looked at the fat bodies under a microscope, they saw even more evidence of this. They found reduced lipid storage, and in some cases they even found remnants of the larval fat body, which should have been broken down and transformed during development.
So it's like their fat body development had been stunted somehow.
Exactly. It seems like that early complex 1 dysfunction interfered with the normal maturation of the fat body.
And that would explain why they were so vulnerable to starvation. They just didn't have the energy reserves to cope.
Precisely.
Okay, that makes sense. Now, what about the NAD plus NADH ratio? I know that's often talked about in relation to mitochondrial health. Did they look at that?
They did. The NAD plus NADH ratio is a really important indicator of cellular metabolism and mitochondrial function.
And it's something that gets thrown around a lot in the health and wellness world, too.
Yeah, and while they did see some alterations in the NAD plus NADH ratio in the flies with developmental complex 1 issues, those changes didn't completely explain the lifespan differences.
So it wasn't the whole story.
Right. It suggests that while the NAD plus NADH ratio is important, other factors are also involved.
Okay, so we've covered the developmental impacts. Now let's switch gears to that really surprising finding about how severely reducing complex 1 in adulthood actually extended lifespan.
Right. So in these experiments, they really hammered the fly's complex 1 function.
Like how much did they reduce it by?
Up to 75%, which is a massive reduction.
Yeah, 75%. I mean, that's got to be pretty debilitating, right?
You think so? But here's the crazy thing. These flies with the severe adult onset complex 1 reduction didn't have shorter lifespans.
They didn't?
Nope. In fact, they actually lived longer under certain conditions.
Longer? Even with their complex 1, practically on life support.
Yeah, it was really unexpected.
Yeah.
And it suggests that maybe there's something beneficial happening in response to that severe reduction.
So it's like the flies are somehow adapting to this extreme mitochondrial challenge.
That's one possibility. And it really makes you rethink the whole idea of mitochondrial decline as being purely detrimental.
So it's not just a passive breakdown. It might actually be an active response, a way for the organism to cope with stress.
Exactly. And that's a really exciting new direction for aging research.
Absolutely.
Yeah.
So how do they try to explain these seemingly contradictory findings? How can a small reduction in complex 1 during development be so harmful while a huge reduction in adulthood is beneficial?
To get that, they took a multiomics approach.
Multiomics, what's that?
It basically means looking at the biology from multiple angles at the same time. So they looked at gene expression, protein levels, and metabolites.
So they were trying to get a comprehensive picture of what was happening inside the flies at a molecular level.
Exactly. And they wanted to see how the molecular profiles differed between the short-lived flies with developmental complex 1 dysfunction and the long-lived flies with the adult onset reduction.
And did they find any differences?
Oh yeah, tons. They identified thousands of genes that were expressed differently between the two groups.
Thousands. Wow. So the molecular landscape was totally different.
Yeah. And what was interesting was that there was a significant overlap in the affected genes between the flies where they knocked down ND18 and the flies where they knocked down ND75.
So regardless of which subunit they targeted, there was a core set of genes that responded to that early complex 1 dysfunction.
Exactly. And when they looked at the functions of these genes, it became clear that a lot of them were involved in metabolism.
Metabolism. Okay, so this goes back to what we were talking about earlier with the fat body and energy storage.
Right. They found disruptions in lipid homeostasis, the transport and metabolism of fats, protein, and sugar metabolism, and translation.
Translation. That's the process of making proteins, right?
Yes, and it's a very energy-intensive process, so it makes sense that it would be affected if the mitochondria aren't working properly.
So basically, these flies with the developmental complex 1 dysfunction had widespread metabolic problems.
Exactly, and these problems were particularly pronounced in certain tissues.
Which tissues?
The fat body, the midgut, and the heart showed the biggest changes in gene expression.
So the fat body again and the midgut, that's part of the digestive system, right?
Yes. And the heart, of course, is crucial for pumping blood and delivering nutrients throughout the body.
So these are all key organs involved in metabolism and energy use.
Right. And the fact that they were so affected in the short-lived flies makes sense given the overall metabolic chaos that was going on.
Okay, what about the metabolomics data? Did that support the gene expression findings?
Absolutely. They found hundreds, in some cases, over 1,000 metabolites that were altered in the short-lived flies compared to the long-lived ones.
Wow. So not just changes in gene expression, but also in the actual metabolites themselves.
Right. And again, the biggest differences were seen in the pathways related to sugar, lipid, and amino acid metabolism.
So that metabolic derangement was showing up at every level.
Exactly. And they even found this interesting thing with the amino acid lysine.
Lysine, what's special about that?
They found an increase in lysine levels and a decrease in the activity of the enzymes that break it down.
So lysine was building up.
Right. And that's actually been seen in other models of complex 1 deficiency, too.
Interesting. So it seems to be a consistent finding.
Yeah, and then the proteomics data basically backed up everything else.
So they saw changes in protein levels that were consistent with the gene expression and metabolite data.
Exactly. And again, the biggest changes were seen in the fat body, midgut, and heart, particularly in proteins involved in lipid protein and sugar metabolism.
So it all comes back to that metabolic dysfunction.
Exactly. It seems like that's the root of the problem in the short-lived flies. And it all stems from that early disruption in complex 1 function that messed up their fat body development.
Okay, so just to recap, even a small hit to complex 1 during development can have huge consequences later in life, leading to metabolic problems, stress sensitivity, and ultimately a shorter lifespan.
Right. Development is clearly a critical window for mitochondrial health.
And this has implications for how we think about mitochondrial diseases in humans.
Absolutely. The severity and timing of mitochondrial dysfunction during development could explain why we see such a wide range of symptoms and ages of onset in these diseases.
So some people might have severe problems early on, while others might not develop symptoms until much later.
Exactly. And it also raises the question of whether some of these developmental problems can be reversed or at least mitigated.
That's a big question, and it's something that researchers are actively investigating.
Yeah, there's some evidence from mouse models that restoring complex 1 function in the brain after birth can have some benefits, but it doesn't completely reverse the damage.
So there might be some hope for interventions, but it's still early days.
Right. Now let's circle back to that other really mind-blowing finding, the fact that severely depleting complex 1 in adulthood didn't shorten lifespan and could even extend it.
This is the part that really challenges our assumptions about aging.
It does. It suggests that maybe we've been too quick to dismiss mitochondrial decline as purely a bad thing.
Like maybe it's not just wear and tear. Maybe it's an adaptive response.
Exactly, and there's growing evidence to support this idea. This isn't the first study to find that reducing complex 1 activity can actually be beneficial under certain circumstances.
So it's not just a fluke. It seems to be a real phenomenon.
Right. And it really opens up a whole new avenue for research into aging and longevity.
So instead of just trying to prevent mitochondrial decline, maybe we should be looking for ways to harness those adaptive responses.
That's a really intriguing possibility.
Okay, so to sum it all up for our listeners, when it comes to complex 1 dysfunction, it's not just about how much, but about when. A small problem early on can have big consequences, but a big problem later in life might actually be a good thing.
Exactly. And it highlights just how complex and nuanced the relationship between mitochondria and aging really is.
And how much more we have to learn.
Absolutely.
Well, this has been a fascinating deep dive. It's amazing how much we've learned about complex 1 and its role in aging.
Yeah, and it all came from those tiny fruit flies.
It just goes to show that sometimes the smallest creatures can reveal the biggest secrets about life.
Absolutely. And for those of you who want to delve even deeper into this research, I highly recommend checking out the original paper by Stefanos and colleagues. It was published in 2025.
It's definitely worth a read.
Yeah.
And it's a great example of how scientific inquiry is constantly pushing the boundaries of our understanding.
Yeah, we're always learning new things and challenging our assumptions.
And that's what makes science so exciting.
It truly is.
Well, thanks for joining us for this deep dive into the fascinating world of mitochondria and aging.
It's been a pleasure.
Until next time, keep exploring, keep questioning, and keep diving deep.